Claudia Shitty, are you ready?
Hello, yes, we're ready.
Okay.
Thank you for the floor. I'll be sharing the screen.
You have 50 minutes to present your work, followed by 5 minutes of the question for the audience. Claudia, you can begin, please.
Yeah, so we have decided to focus on creating an adaptive network model which simulates the role of the gut microbiome on the circadian cycle, which is the 24-hour body clock, on mood and on insomnia, which is the sleep disorder where you struggle to fall asleep, through the gut-brain axis. Next slide, please. So the microbiome is the population of microorganisms that live on your body. And here we focus specifically on those in the gastrointestinal tract. So the composition of this microbiome affects different biological processes in your gut. So these can then travel along the microbiome-gut-brain axis using neurotransmitters, hormones, or inflammatory cytokines. And these then affect the biochemistry of your brain, in turn affecting mood, circadian rhythm, and insomnia. Next slide, please. There's 3 main pathways which are part of this microbiome-gut-brain axis. So there's the neuroendocrine pathway, the immunoregulatory pathway, and the vagus nerve pathway. But we decided to focus on the neuroendocrine and immunoregulatory pathways because we wanted to focus on the endocrine and immune systems and how these work to regulate different hormones, which are important for a regulated sleep cycle. So we've got the neuroendocrine pathway on the left, and this is where the endocrine cells in the gut are stimulated by these microorganisms of that gut microbiome. Depending on what these microorganisms are, it depends on what these what molecules these bacteria are secreting. And dependent on that, different neurotransmitters are released that then travel along the bloodstream, along the HPA axis, which is the hypothalamic pituitary adrenal axis. This pathway coordinates the body's stress response, and it does this through coordinating cortisol regulation, cortisol being the body's main stress hormone. So tryptophan metabolism is also regulated through this pathway. Tryptophan is a precursor of the hormones serotonin and melatonin, which are the 2 main sleep-regulating hormones. We then have the immunoregulatory pathway, which is the pathway to the right, where immune cells in the gut detect pathogenic microorganisms, which would cause these cells to secrete pro-inflammatory cytokines. And these cytokines travel along the bloodstream and can cause inflammation all over the body. Sometimes these pathogenic bacteria can produce neurotoxic metabolites. This travels along the blood to the brain and can cause lots of damage there to different molecular processes, which can affect mood, circadian rhythm, and can increase the chances of insomnia or worsen insomnia that the person would already have. This can cause inflammation in the gut, so you can get a leaky and inflamed gut, meaning that these toxins released by the bacteria pass from the gut and then into the blood. Next slide, please. So if you want a healthy microbiome to reduce your chances of insomnia or improve this condition, you can try to reduce your stress levels, have a healthy diet, maybe take some probiotics, because lower stress means lower cortisol levels. And when cortisol is too high, this can dysregulate your circadian rhythm. This also regulates the clock genes. Clock genes encode the proteins that regulate that circadian rhythm. So we chose to focus on two probiotics, Bifidobacterium and Lactobacillus, because these are both found naturally in a high concentration in your gut microbiome. They have very beneficial effects in upregulating serotonin and melatonin production in your body. If you have more melatonin and serotonin, then generally insomnia is improved. The current drugs on the market for insomnia have lots of side effects and are not very effective over the long term. So research into probiotics to treat this condition is definitely worthwhile. Next slide, please.
All right, so these are the 30 states that we used for our network simulations. On the left side in the pink, you can see all of our states for the bacteria that we're researching, the various neurotransmitters like cortisol, serotonin, melatonin, states for insomnia and stress. And on the right side in the blue and the purple, we have our adaptation states that correspond to our neurotransmitters and stress to better reflect neuroplasticity. So in the blue, we have adaptation states for connection weights, and in the purple, we have adaptation states for speed factors. Next slide, please. And here is our adaptive network model. You can see that it has 3 layers. The top 2 layers, the first order and second order reification levels, are our adaptation levels. Next slide. And here are the combination functions we used in our model. So the identity function was used for states that do not change in their levels, such as the stress, the external stress that the person is undergoing. Alogistic or advanced logistic sum is used for states that change in general. So this is used for the majority of our states. And Hebbian learning is used for adaptivity and our adaptation levels, such as the connectivity weight and the speed factors. And bounded growth, we use this for the states that represent bacteria because we wanted to better reflect how bacteria can often grow in the gut microbiome. Next slide.
So in my section, I'll be describing in detail which specific scenarios we decided to model in the dedicated software environment in MATLAB with respect to our domain, our intended expectations from the simulations we ran, and our final inferences from the graphs generated. For the sake of simplicity, we ignore the first and second order adaptive states for the purpose of analysis. So in our first state, we have low levels of probiotics and high levels of bad bacteria in the gut. So as we have achieved this by taking initial values for probiotics as 0, and for the bad bacteria in the gut, we have used the bounded growth function, and it starts from 0.8 as the initial value. So as we can see that we intended to look at the effects produced on the system state of insomnia, which is X15 state over here, and its corresponding effect felt as sleep deprivation, which is our X16 state, in the scenario of imbalance of bacteria, such that we have no probiotics, which is our lactobacillus and bifidobacteria, but there is high levels of bad bacteria, which are the pathogenic bacteria such as Clostridium perfringens, Staphylococcus, etc., that release toxins and or trigger disease in the system. So this scenario is what we term as dysbiosis. As expected, we can see that the X4 state, which is our immune cell, it increases, and to bring the level of bad bacteria in the gut down. This causes gut permeability to increase, where this bad bacteria then permeates into the bloodstream. EEC1, which is our first enteroendocrine cell pathway, which activates HPA1 axis, is not activated, but there is an effect that is felt, which brings the levels of cortisols up, and the perceived state of stress that is felt by the body, as we would have expected in the normal situation. Next, we have our scenario 2, where we have high levels of probiotics, but low levels of bad bacteria in the gut. This is the opposite of our first scenario. And we have done the same, where we have kept the bad bacteria at a 0 level throughout. And our probiotics, lactobacillus and bifidobacteria, show bounded growth. I'm sorry, I think my computer hanged. I'm sorry for. I'm sorry, because of my low battery, I cannot share anymore. Can somebody share screen?
I can share the screen, just give me 1 second.
All right, thank you so much. Our intention in this situation was to find how the bacteria react in independent situations where they're not reacting with each other at the same time. So the first HPA state, which is activated by the lactobacillus, then acts, has a negative impact on the HPA axis, which brings down the cortisol levels and the effect of stress, and in turn, the level of insomnia and sleep deprivation. In a similar manner, the bifidobacteria acts on the HPA2 axis, which releases serotonin, and it also acts on the tryptophan, which produces melatonin. This also brings down our insomnia and sleep deprivation levels and leads to the clock gene regulation. Next slide, please. So lastly, we have run a scenario where there's low levels of probiotics and low levels of bad bacteria in the gut, which means that we have kept our all our bacteria at a level of initial value of 0 throughout the runtime. This leads to a state which is termed as dysbacteriosis, but this is not a usual situation because we usually have lactobacillus and bifidobacteria in the gut. But in case they are absent, the immune response is not activated. The cytokine response is triggered due to the constant external stressor factor, which is at a constant of 1. Throughout, this raises cortisol production, and the system then has, next slide, please. The system then has a perceived state of insomnia, but lower than in the case of where there's bad or pathogenic bacteria in the blood as well. Next slide, please. So in conclusion, we'll be discussing some of the future perspectives that our model and our research could have in further research. So first, there could be bacteria in competition where good and bad bacteria both compete for food and resources to grow and proliferate in the same home environment, which is the gut, which then could have implications for mood and sleep. Next is real-world implications, where real-world situations such as night shifts or eating disorders or jet lag and such could be modeled. There could also be a 1-1-1 state where all the 3 types of bacteria are active and interacting in the same home environment. We have kept the external stressor value at 1 throughout. There could also be different external stressor values, which could then have their own functions for growth. There is also a strain of bacteria called opportunistic bacteria that unlike bad bacteria, do not attack the system immediately, but they wait for the immune cells the immune system of the body to reach a low enough state, and then they attack and release cortisol, which then increases stress and insomnia. So I think our model and research was a simplified and organized effort into modeling bacteria present in the gut microbiome and their role on the overall mood, sleep-wake cycles, circadian rhythm, and any resultant sleep disorders in the person, such as insomnia. We are very happy to present at BICA. Thank you. Any questions, please?
Thank you, Sulagna, Claudia, and Kinley for your participation. Now we will proceed with the questions. If anyone has any question, please raise your hand, please.
I have a question.
Okay.
Hi. A system like this that you're modeling here is quite complex, and there are many chain reactions that are connected to each other. And my question is, how can you validate if the simulation data that you are generating with your model can be really what is happening in real life? Do you have a standard procedure for doing this validation, or you just simulate and need to believe in the what the simulation data provides you?
So we did qualitatively validate, but we didn't numerically validate because we have not yet found enough data to be able to do so on this topic. So what our model is based off of is just research papers discussing conclusions between the effects of like one hormone on a certain process. And then we built it up around lots of different research papers on these topics. So we would definitely need to numerically validate this model so that we would have a greater accuracy and make it more realistic. But that was what we could do with our time so far.
But it would be interesting, for example, if you, even on a qualitative way, if you can connect saying, okay, for example, study A and B reviewed the such and such kind of phenomena, and this is what is happening in the simulation. The other study provides such and such, and this is what we are having in our simulation, because this will give credibility to the model that you're building. So I think it would be a good suggestion to, even if you don't have a quantitative validation, you can validate it qualitatively by presenting the studies that you use to show that your process is doing what is expected to do. Okay, just a suggestion.
Definitely, if we had more time for a presentation, that would definitely be a good thing we could include.
Thank you.
Thank you.
Is there any other question? Okay, Howard, please.
Yes,
Question.
hello. 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 Hi, Ricardo. It's nice to hear you. I don't think I can see you, though. I thought that was a good question. It's an interesting topic, the probiotics. We've known for about 20 years that they're quite effective there's double-blind controlled studies now for depression, for insomnia, for irritable bowel syndrome, which actually is part of psychiatry. So, a lot of psychiatric issues, but also for colitis, Crohn's disease. But somehow it hasn't made it into mainstream medicine just because it takes so long. And I guess there's, there may be not as much profit potential as like in other pharmaceuticals. However, but there, I think your question, Ricardo, is a good question. It's so hard to actually do trials, which is why, it's not part of mainstream medicine. If I see people that have depression and I give them antidepressants, that's great. If my recommendation was probiotics, people would say, what's going on? But I think the problem is it's really hard to do the studies, although I think it'd be interesting doing meta-analysis. People have done it in other fields. There's somebody, Cipriani, who's done it for antidepressants, comparing all sorts of situations, which it would just be too hard to do. The trials cost many millions of dollars to start figuring out what's going on. But I thought it was a good talk. I thought it was interesting, a nice system that may have some utility. A very nice talk.
Thank you. Yeah, I agree that this could be taken a lot further. We just need to find, some good amount of data to be able to progress our model on this. But it's a really interesting topic, I agree.
Yeah.
Okay, thank you for your presentation.